Hello everyone, welcome to Health Watch Ismanla. In this episode, I will talk about how cytokine storm is related to COVID-19 illness and COVID-19 symptoms. So we know that we are going through one of the worst pandemics caused by SARS-CoV-2 virus or coronavirus, and more than 370,000 people have already died due to this virus, and we know that 95 to 96% of people recover from this sickness, and among this 20 to 30% of people show acute respiratory distress syndrome, or you can say respiratory failure, and this symptom is associated with an increase in inflammation or cytokine storm. So in this episode I will try to explain how cytokine storm is associated with respiratory failure. And also you must know that when you have a lot of inflammation, there is lung damage, and at certain stage, if the lung damage is massive and the patient is experiencing severe symptoms, the patient can go into acute respiratory distress syndrome or respiratory failure. We'll try to explain where in the lungs this occur. So we have something called alveolus or alveoli. So we have our airway here quickly and trachea, and this trachea then branches into something called this is your right lung, something called a bronchus, and then bronchus is further branches into bronchioles and then ends up into a sac called air sac or alveolus. And this alveolus, if you magnify, it may look like this. It's just for the schematic representation. And this play a very important role in gas exchange, and that's how when you take the air in, and you will have oxygen, and it will end up into the air sac, and we have oxygen here, and that goes into the blood vessel. That means each alveolus is surrounded by blood vessels. Carbon dioxide in the blood will go into alveolus, and then oxygen in the alveolus will then go into the blood, and that's how the supply of oxygen is continuous in our body. So one thing that I will tell you here about is epithelial cells. So the epithelial cells play a very important role because the inner lining of the airway, starting from your nasopharyngeal cavity to your alveolus, is all covered by epithelial cells. And if you look at the alveolus, there's epithelial cells, and these are also called type 1 and type 2 epithelial cells. And these epithelial cells, for example, this is your type 2 and this is type 1, just for the sake of information, and they represent 99% of your lung inner surface area. Here, because as I said, they cover the almost entire inner surface area of the lungs, they become the major target for viral infection. So what happens is, here, if you have the virus infection, and it can infect upper respiratory also, and it may go down and end up in coming here, but the viruses are the simplest of the living organism, and they cannot just multiply when they come in. They have to get into a host cell, so that they have to multiply. So if you look at this, the host pathogen interaction, so everything occurs at the cell level now. So if you look at this in a little bit in more detail, so this is your epithelial cells being attacked by virus, and here, for example, the epithelial cells here express S2 receptor, which is important for the coronavirus to bind, and the coronavirus can then, through its spike protein, also called S protein, can bind to S2, and that's how they attach to the host cell, and it can enter into the host cell. And immediately after entering into the host cell, they release their genomic material. So every virus will have their genetic material coated inside, and being a living organism, genetic material is very important. So then that's how then they here hijack the host machinery. So as I said before, so viruses need host machinery for their own multiplication and replication, making more progenies. So here it will require the host factors and make more of its own genetic material, and further it can utilize the host compartments here and then make more viruses. That's how then it can come out. So if one virus enters into the cell, hundreds and thousands of viruses come out of the cell. The host cells, they do not just stand there, and let the virus go in and get away with multiplying themselves. So hosts have also developed very sophisticated machineries and electrical sensors, and these sensors can sense the viral RNA or viral proteins, particularly viral RNA, can initiate and then cascades of signaling, and this signaling can of course activate genes in our nucleus, and by the process of transcription and translation, these are the biological processes through which we make more proteins when we get a stimuli or signal from outside, and then this can, through these processes, the cells can make more proteins and cytokines or chemokines. Now if you see, these are the earliest or the initial response against the viral entry from the host side, and this happens as soon as the virus enters, and by the time 8 hours or 9 hours or 1 day after infection, you will have this uh, reaction occurring here. So now if you zoom out and come back here, now this virus here goes inside, and upon entry the host senses the virus and produces cytokine in the milieu. Now what they do, these cytokines and chemokines, so they can activate the epithelial cells and also other immune cells. There are resident macrophages and dendritic cells in the uh, alveolus or the airway, and when the cytokines then activate other epithelial cells, and they become resistant to further infection, and so that these are the kind of first response or the first protective response that host is trying to prevent itself by from the viral replication. And another important thing is the chemokines can go into the blood, and both cytokines too, but chemokines are the kind of messenger proteins. They can send signals outside of the lung, and by through blood it can recruit more immune cells to the site of infection. So right now you have infection going on here, you can call it site of infection, and this will soon turn into site of inflammation, and that's I will tell you how. So now for example, the you have an initial mild response here producing chemokines, and these chemokines send the signal, and we have our bone marrow, our bone, and inside the bone there is marrow or bone marrow. Bone marrow is a kind of factory for immune cells. It will continuously produce immune cells, and these are ready to come in or ready to respond to the signals wherever there is some kind of injury or infection. So basically once they get the signal, and these cells will come into the circulation. And what are the immune cells in the bone marrow? Majorly they are neutrophils, and these are other cells are like monocytes, eosinophils, So the first responder here is neutrophils. They represent like 40 to 50% of your whole immune cells in the bone marrow, and they can immediately get recruited to the site of infection here. And the role of neutrophils is really not very clear in viral infection, and it is understood that they actually do more harm to the tissues than protecting. But in case of bacterial infection, because bacteria are mostly, some of the bacteria are outside the cells, they can directly capture the bacteria if there is a bacteria, and that's how the neutrophils are very important for bacterial infection. And after a neutrophil, then monocytes come in, and these monocytes are also very important in promoting inflammation. For example, NK cells also come in here at the site of infection, and these NK cells have a unique property where they can directly identify the infected cells. For example, this cell is infected here, and NK cells here can directly identify the cells which are infected by the virus through the expression of certain proteins on the surface or not expression of certain proteins on the surface, and they can directly kill the cells which has the viral replication going on, so that there is no more virus growing in the cell. But overall, there are so many other immune cells fall in or gets crowded at the site of infection. As I cannot go through each and every detail here, I will try to show you a graphic here, and this is from an experimental model where you see so many immune cells that we can study about each and every cell and what they are doing. But here these immune cells then produce a lot of cytokines. Now in response to the infection, they kind of get panicked, and then these produce these cytokines and chemokines, and they also go systemic, these cytokines. And there are two things here now. One is this site of infection becomes now site of inflammation, and second thing is if the inflammation is really massive, it can also be associated with then when you are having fever here, because your body is really going through so much of trying to control the inflammation, and at that point your body temperature also raises, probably in order to control the virus replication by heating up the body. And second thing is now when you have a massive inflammation, I will talk a little bit about moderate inflammation or massive inflammation or good inflammation or bad inflammation. So you have an immune cell here, and for example one of the cells here, and here are so many cytokines in the milieu, and it can, for example, the cytokine can bind to its receptor and amplify more signaling. Basically the cells get activated by producing more cytokines and chemokines and other factors that contribute to the inflammation. So overall, so if it happens, or the process of inflammation and production of chemokine is moderate, it may help for the body and it may help in controlling the virus. But then depending upon individual, it could be genetic, environmental, or it could be the kind of immune profile. For example, if someone has already some kind of infection before the viral infection, I would not say infection, or I would say a different immune profile. Say for example, if you have allergy, you will have a lot of eosinophils in your lung that may or may not help you, but it can certainly change your inflammation. For example, first of all, I will here go through some of the cytokines that are important here are IL-6, TNF alpha, IL-1 beta and IL-17 was observed in many of the COVID-19 patients, and these cytokines are majorly present in during the cytokine storm. And for example, here IL-6 can then bind to other immune cells and amplify, expand the inflammation. So at one point, suppose these cytokines, there are a number of chemokines and other proteins too. It's just for the sake of simplification. So there are a number of factors why a moderate inflammation, or in other words, how a moderate inflammation could turn into a cytokine storm. So now the next important thing that I will go through is how the inflammation itself now causes tissue damage. So let's look at this another drawing, another alveoli. So as I said that we have a lot of inflammation going on here and a lot of cytokines here, that results into either due to the inflammation or due to the viral replication, the epithelial cells here starts dying or die, and and then you will have a lot of dead cells here, definitely. Now it is also depends to what extent and how many alveoli are like what extent of damage you have in the lungs that can result into uh, the immunological event then turn into a symptom. For example, if you have a lot of cells dying, then you can suffer or experience cough, because cough is a symptom where it's one of the mechanism, or one of the body's approach is to remove the debris and dying cells. And another thing is, now we know that the alveoli are associated with blood vessels, and these blood vessels under massive inflammation can break or rupture, because you have a breakage of capillaries or blood vessels, and there could be leakage. So it could be leakage of proteins, and blood is full of serum proteins, and these serum proteins can then come into your lung or alveolus. And also, now if you look back here, we had damage, and then we have due to the damage we will have leakage of both proteins. And in experimental condition or animal models, you can directly look at these proteins in the lungs, and that would be indication of your lung injury. And another thing is, then with the leakage of proteins, then you have also accumulation of fluid. And the third thing is then here accumulation of fluid. So this accumulation of fluid then results into interruption of your gas exchange. So now you know that when you don't have an infection, otherwise a healthy lung, you will have a very intact gas exchange here, particularly oxygen going into the blood, and then carbon dioxide here is going into the alveolus. And this gas exchange, when you have already leakage and with the alveolus filled with fluid, can completely interrupt it. So you no longer have gas exchange, and that can lead to what? Reduction in oxygen. Now definitely, of course, this is depend upon like how many alveoli or what extent of damage the lung has. But if it is massive, or in conditions like acute respiratory distress syndrome or respiratory failure, your oxygen saturation of blood will go down. That's why when you go to doctor to check your lung function, they can check your oxygen saturation. So with reduction in then oxygen in the blood, and one thing then can happen here is you can experience shortness of breath, because your body is trying to compensate for the loss of oxygen here in the blood. And that's how these immunological events are associated with, possibly associated with the symptoms. And with that, I will stop here. And this is a quick scientific explanation how the cytokine storm could be associated with lung damage, and how the host immune cells can sense the virus, and how it can kickstart the immunity and particularly inflammation. So with that, if you have any questions or comments, please write down. I will try to address it, and also don't forget to subscribe the channel. Thank you.